Hello viewers, welcome to ARC. The topic for this video is bacterial anatomy. The structure of an idealized bacterial cell shows an outer layer which consists of two components, a rigid cell wall and a cytoplasmic or plasma membrane. The components are cell interior, which are cytoplasm and cytoplasmic inclusions, and additional structures which are flagella and fimbriae. The outermost layer of the bacteria is cell wall. The cell wall accounts for the shape of the bacterial cell and confers on its rigidity and ductility. The cell wall cannot be seen by direct light microscopy and does not stain with simple stain. Structure: the bacterial cell wall are 10 to 25 nanometer thick. Chemically, the cell wall is composed of mucopeptide scaffoldings which are formed by N-acetylglucosamine and N-acetylmuramic acid molecules altering in chains which are cross-linked by peptide chain. In general, the wall of gram-positive bacteria have a simpler chemical nature than those of gram-negative bacteria. The cell wall carries bacterial antigens that are important in virulence and immunity. Cell wall of gram-positive bacteria. It contains two layers. The first one is peptidoglycan layer and the second one is teichoic acid. The first layer is peptidoglycan layer. This is thick than that of gram-negative bacteria. Teichoic acid. Teichoic acid is the major surface antigen of gram-positive bacteria, which contains ribitol or glycerol polymers. The cell wall contains a significant amount of teichoic acid. The two types of teichoic acid are present: cell wall teichoic acid, which is linked to peptidoglycan, and the membrane teichoic acid, which is linked to membrane glycolipid. This is the diagrammatic representation of the cell wall of gram-positive bacteria, which can be seen in three layers: capsule, cell wall, and cytoplasmic membrane. Coming over to cell wall of gram-negative bacteria. Lipopolysaccharides, these are present on the cell wall of gram-negative bacteria, which account for their endotoxic activity and O antigen specificity. Outer membrane. The outermost membrane of gram-negative bacterial cell wall is called outer membrane, which contains various proteins known as outer membrane protein. Lipoprotein. It attaches the protein of peptidoglycan to lipid of outer membrane. Peptidoglycan. It is thin and is bound by lipoprotein and plasma membrane. This is the diagrammatic representation of cell wall of gram-negative bacteria, which is divided into three layers: capsule, outer membrane, which is also called as cell wall, and the inner membrane. The cytoplasmic membrane or the plasma membrane. The cytoplasmic membrane is a thin layer lining the inner surface of the cell wall and separating it from the cytoplasm. It acts as a semi-permeable membrane controlling the flow of metabolites to and from the protoplasm. Chemically, the membrane consists of lipoproteins with small amount of carbohydrates. Cytoplasm. The bacterial cytoplasm is a colloidal system of the variety of organic and inorganic solutes in a viscous watery solution. The cytoplasm stains uniformly with basic dyes in young culture and become increasingly granular with age. The cytoplasm contains ribosomes, mesosomes, inclusions, and vacuoles. Ribosomes. These are the centers of protein synthesis. They are slightly smaller than the ribosomes of eukaryotic cell. They are seen integrated in linear strands of mRNA to form polysomes. Mesosomes or chondroids. These are vascular, convoluted, and multilaminated structures formed as invaginations of plasma membrane into the cytoplasm. They are more prominent in gram-positive bacteria. They are the principal sites of respiratory enzymes in bacteria and are analogous to the mitochondria of eukaryotes. Intracytoplasmic inclusions. These may be of various types, the chief of which are volutin, polysaccharide, lipid, and crystal. They are characteristics for different species and depend on the age and condition of the culture. Volutin granules are highly refractive, strongly basophilic, consisting of polymetaphosphate. Volutin granules are characteristically present in diphtheria bacilli. Nucleus. Bacterial nuclei can be demonstrated by acid or ribonuclease hydrolysis and subsequent staining for nuclear material. Bacterial nuclei has no nuclear membrane or nucleolus. The genome consists of a single molecule of double-stranded DNA arranged in the form of a circle. The bacterial chromosome is haploid and replicates by simple fission instead of mitosis as in other cells. The difference between nuclei of bacteria and that of other organisms form the main basis for classifying them as prokaryotes and eukaryotes. Bacteria may possess extra nuclear genetic elements consisting of DNA. These cytoplasmic carriers of genetic information are termed as plasmids or episomes. Flagella. Motile bacteria except spirochetes possess one or more unbranched long filaments called flagella, which are the organs for locomotion. Structure. Each flagellum consists of three distinct parts: the filament, the hook, and the basal body. The filament is external to the cell and connected to the hook at the cell surface. The hook basal body portion is embedded in the cell envelope. Flagella are made up of proteins similar to keratin or myosin. Flagellar antigens induce specific antibodies. The presence or absence of flagella and their number and arrangement are characteristic of different genera of bacteria. This is the diagrammatic representation of general structure of the flagellum, which contains three parts: filament, hook, and the basal body. Fimbriae. Some gram-negative bacilli carry very fine hair-like surface appendages called fimbriae or pili. They are shorter and thinner than flagella and project from the cell surface as straight filaments. Pili comprise self-aggregating monomers of pilin. Fimbriae can be seen only under the electron microscope. They are unrelated to motility and are found on motile as well as non-motile cells. Fimbriae function as organ of adhesion, helping the cell to adhere firmly to particles of various kinds. A special type of fimbriae are sex pili. These are longer and fewer in number than other fimbriae. They are found on the male bacteria and help in attachment to those cells to the female bacteria, forming hollow conjugation tubes through which it is assumed genetic material is transferred from donor to the recipient cell. Thank you. If you have any queries, comment us below, and also please like, share and subscribe. Thank you.